We're now going to start the microbiology module, and um, book says that microbiology questions on the step one exam often require two steps. Given a certain clinical presentation, you will first need to identify the most likely causative organism, and you will then need to provide an answer regarding some features of that organism or relevant antimicrobial agents. For example, a description of a child with fever and a petechial rash will be followed by a question that reads, from what site does the responsible organism usually enter the blood? This section therefore presents organisms in two major ways: the individual microbial profiles and in the context of the systems they infect and the clinical presentations they produce. You should become familiar with both formats. When reviewing the systems approach, remind yourself of the features of each microbe by returning to the individual profiles. Also, be sure to memorize the lab characteristics that allow you to identify microbes. We're going to cover basic bacteriology in this section. So first of all, there are bacterial structures, and the first structure is the appendages, their chemical composition, and their function. So the first structure is flagellum. Flagellum is composed of proteins, and its function is motility. There we go. The second structure is pilus or fimbria, and they are composed of glycoprotein, again a protein, but this time a glycoprotein, and they mediate adherence of bacteria to cell surface. And sex pilus forms during conjugation. We're going to discuss conjugation further, but they are mainly for the adherence. Then we have specialized structures called spore, which has a keratin-like coat, dipicolonic acid in amino acid in its molecule, and peptidoglycan being the structure core, and a DNA being the material that is present inside the spore as the nucleus containing material. Its survival is that it resists dehydration, heat, and chemicals, and it also resists all kinds of heavy metals and antibiotics mostly. So dipiclonic acid is an important thing. Dipicolinic acid. So here's an endospore. To study its structure, there is a small acid-soluble protein inside it, its core, inner membrane, cortex, outer membrane, and spore coat. And but where is my dipicolinic acid? It's a structure here, but uh, spore. Mm, it's part of the structure of the spore, basically. So, dipicolinic acid, but peptidoglycan, yeah, I think a better question would be dipicolinic acid role in spores. It is an amino acid. In bacterial spores, it is a key molecule that ensures dormancy, extreme resistance, and DNA protection by forming a calcium BPA complex in the spore core. Dipicolinic acid is concentrated in the core of bacterial spores, particularly in Bacillus and Clostridium species. So these two are the core spore-forming bacteria, and constitutes roughly 5 to 25% of the spore's dry weight. All right. Its structure with two carboxylic acid groups and nitrogen atom allows it to act as a tridentate ligand, forming strong bonds with divalent cations, primarily calcium ion, resulting in a calcium DPA complex. Okay. So moving on. Then we have the cell envelope. There is a structure of the cell envelope. This is a gram-positive cell envelope, and this is a gram-negative cell envelope. Um, First of all, there is a cytoplasmic membrane or the cell membrane. Then there's these raw chip peptidoglycan wall, which is thicker in gram positive and thinner in gram negative bacteria. Um, but in the gram positive bacteria, between the peptidoglycan wall, there is this pink structure which is known as flagellum. We just discussed that This flagellum is for motility and it is made up of proteins and is also present here in the gram-negative. It is common to both, this flagellum. Then there is this green structure which is known as the pilus and it is also common to both. And then there's this coverton right here which is known as the capsule which is also common to both. Now, what is unique to the gram-positive is lipoic acid right here. And what is unique to the gram-negative is the endotoxin or lipopolysaccharide and the porin protein that forms the outer membrane. Okay, so this whole outer membrane is not present in the gram-positive, and this whole outer membrane is made up of lipopolysaccharides or LPS, which contain endotoxins, and this protein known as porin. And there's a white periplasmic space containing beta-lactamase, which is very important. Now let's discuss the reading. The capsule is a discrete layer made up of polysaccharides and rarely proteins, and it protects against phagocytosis. Okay, so what I've read about the polysaccharides is that a specific component of these polysaccharides decides the serological type of the bacteria, and it is very important for developing antigenicity or a serologic type to particular bacteria. And it is made up of polysaccharides and rarely proteins, and it protects against phagocytosis. It is also important for adherence, for identification, for determinants of virulence, and for determinants of which vaccines are to act based on whether the bacteria is capsulated or encapsulated. Then we have a slime layer, which is a loose network of polysaccharides. Again, it mediates adherence to surface, plays a role in biofilm formation, in welling catheters, and prosthetic wall attachment. All this is mediated by the slime layer. They have not mentioned it here. The slime layer is actually produced by some bacteria and not produced by others. The outer membrane has an outer leaflet. This part, an outer leaflet that contains the endotoxin or LPS and LOS, and it has embedded proteins, which are porins, and other membrane proteins such as OMPs, and it has an inner leaflet made up of phospholipid. It is present in gram-negative only. Endotoxin is lipid A, induces TNF and IL-1, antigenic O polysaccharide component. Most OMPs are antigenic and porins transport across other membrane. Okay, so this LPS is basically composed of three parts. A lipid A molecule, which is mentioned here, that induces TNF, tissue necrotic factor, and IL-1, and an antigenic O polysaccharide component that is recognized by the antibodies, and the antibodies bind to this O region, and there's also another O polysaccharide region. Most OMPs are antigenic. I'd like to see what are OMPs. OMPs and there you go. Okay, so outer membrane proteins. Most outer membrane proteins are antigenic, and porins are transported across the outer membrane, right? So, porins and other outer membrane proteins. The outer layer is composed of lipopolysaccharide, and lipopolysaccharide has an endotoxin, and lipid A. It is composed of lipid A and phospholipid that extends outward, and O polysaccharide component and a normal core polysaccharide component. And the outer leaflet also has porins and other outer membrane proteins. The inner leaflet has phospholipids. Then is periplasm, which is a space between cytoplasmic membrane and outer membrane in gram-negative bacteria, and there's peptidoglycan in the middle. This is the periplasmic space between the cytoplasmic space and the outer membrane in the gram, and the peptidoglycan layer is in between the periplasmic layer. It accumulates components exiting the gram-negative cells, including hydrolytic enzymes, example beta-lactamases. Then we have the cell wall. Peptidoglycan is a sugar backbone with peptide side chains cross-linked by transpeptidase. It has an endless structure that gives rigid support, protects against osmotic pressure and damage, just like all cell walls do. And the important thing is that it is made up of peptidoglycan, and the capsule was formed of polysaccharides. The cytoplasmic membrane is phospholipid bilayer sac with embedded proteins, example penicillin binding protein and other enzymes. Lipoechoic acids present only in the gram-positive bacteria extend from the membrane to the exterior. This is the inner membrane or the cell membrane that is present in both of these cells, and it has proteins which are also, for example, the penicillin binding protein and other enzymes. And lipoechoic acid that is only present in gram-positive extends from the membrane right here to the exterior. It is a site of oxidative and transport enzymes. Penicillin binding proteins involved in cell wall synthesis, and the lipoechoic acids induce TNF alpha and IL-1. Okay, so the lipid A in the endotoxin in the outer membrane of gram negative was inducing TNF and IL-1. Contradictory to that, in the gram positive cells, the lipoechoic acid is inducing TNF alpha and IL-1. Okay, moving on. We have the stains. The gram stain, it is a first line lab test in bacterial identification. Bacteria with thick peptidoglycan layer retain crystal violet, gram positive, and bacteria with thin peptidoglycan layer turn red or pink, gram negative with the counter stain. These bugs do not gram stain well. These little microbes may unfortunately lack real color but are everywhere. Okay. So these little microbes may unfortunately lack real color but are everywhere. Treponema and Leptospira, they are too thin to be visualized. These little Treponema and Leptospira, too little to be visualized. Microbes, mycobacteria, cell wall has high lipid content. This is highly repeated that mycobacteria has a high lipidosterol content, and that is why it usually takes an acid base stain and not our usual gram stains. So these little Treponema, Leptospira, and mycobacteria. Then we have unfortunately mycoplasma and ureaplasma. Both have plasma in common. They do not have any cell wall, and thus they cannot be stained because cell wall is having the thick peptidoglycan layer that would retain the crystal violet dye, or the thin peptidoglycan layer that would turn red or pink. But mycoplasma and ureaplasma do not have cell wall, so they're not going to take any color. These little mycobacteria, mycoplasma and ureaplasma, microbes may unfortunately lack real. Okay, so the next is the same category, lack real color but are everywhere. Legionella, Rickettsia, Chlamydia, Bartonella, Anaplasma, Ehrlichia. They're primary intracellular. Also, Chlamydia lack classic peptidoglycan because of decreased uramic acid. Okay, so Legionella, Rickettsia, Chlamydia, Bartonella, Anaplasma, and Ehrlichia are basically intracellular, so they cannot be stained. Next is Giemsa stain. We ought to see what that is. Giemsa stain. That's big. Okay. It looks somewhat like this. Um, Giemsa powder, glycerol, and methanol is present inside it, and this is how it stains. These are kind of purple, aren't they? So the organisms it is going to stain are health. Certain bugs really try my patience. H. pylori, chlamydia. Chlamydia was also right here in the not staining one, and it is also right here in um, the one stained by Giemsa. Then there's Borrelia, Rickettsia, again that list of intracellular bacteria that do not take the same, but they are taking Giemsa stains supposedly. Chlamydia and Rickettsia, and there are also trypanosomes and Plasmodium. Okay, so these are trypanosomes. They take the Giemsa stain and the Plasmodium. So H. pylori, Chlamydia, Borrelia, Rickettsia, trypanosomes, and Plasmodium, all of these take the Giemsa stain. Then we have the periodic acid Schiff stain. It stains glycogen, mucopolysaccharides, and it is used to diagnose Whipple disease, which contains the Tropharema whippleyi. So it is very specific for this disease, which is a Whipple's disease, Tropharema Whipple. Oh, I suppose I want you to take a right guess. Whipple. This organism, Toparima whipple, it takes the acid, uh, periodic acid keep stain, and this stain basically stains glycogen and mucopolysaccharides and all that. So, acid base keep stain. It's not acid base, right? It's periodic. I keep forgetting it's periodic acid, yes. Um, it is used to detect polysaccharides and mucosubstances in tissues. It works by oxidizing into glycol groups and polysaccharides, resulting in aldehydes that react with Keeps reagent. Um, I suppose it's sheaves, not skeeves, producing a characteristic magenta color. Okay, so this stains magenta. The glycogen and the mucopolysaccharides are stained into a magenta color, and it is used to diagnose the Whipple disease, which is present right here, and it is caused by the Topherema whippleyi. Then we have the Zeal-Nielsen stain or carbolfuchsin. It is an acid-fast bacteria that is used to stain mycobacteria right here, and Nocardia stains the mycolic acid in cell wall. Both Mycobacteria and Nocardia acid-fast bacteria and Zeal-Nielsen stain or carbolfuchsin is going to stain them, and it stains mycolic acid, which is present in the mycobacteria well, and protozoa, which are an exception right here, are also stained by Zeal-Nielsen stain. The specific protozoa are Cryptosporidium oocyst. So mycobacteria here, Tropharema whippleyi right here, and which was this one? Trypanosomes right here. Trypanosomes, um, Tropharema whippleyi, and mycobacteria. We've done GIMSA, periodic acid sheaf stain, and Zeal-Nielsen stain of carbolfuchsin up till now. There's also a stain called auramine rhodamine stain that is more often used for screening. It is inexpensive and more sensitive. Then we have India ink stain, which stains Cryptococcus neoformans. This structure. It is mucicaramine. Mucicaramine can also be used to stain thick polysaccharide capsule red. Okay, so there are so many stains right here. Mucicaramine stain. Yeah. It is a histological technique used to detect acid mucins in tissues in the capsule of certain fungi, producing a pink red coloration. There it is, this pink red color, and they are saying that it's mucicaramine is used to stain thick polysaccharide capsules red. And India stains are staining this molecule, Cryptococcus neoformans, and mucicaramine are used to stain thick polysaccharide capsules. I don't see the correlation here, but because this is completely a black background against this India ink, but you have to go with it as it is. Next, there is silver stain, Helicobacter pylori, Legionella, Bartonella. Okay, so Legionella and Bartonella were right here. These two. Helicobacter pylori was also being stained by gypsum stain, and it is also being stained by silver stain. Bartonella henselae. Okay, so that Bartonella was just Bartonella, and this one is Bartonella hensela. And fungi, example, coccidioids and pneumocystitis girovicae and Aspergillus fumigatus. These are being stained by the silver stain. Helicopters are silver. Helicobacter pylori, Legionella, Bartonella hensela. Okay, they've not made a mnemonic for that. Coccidioids, P for pneumocystis, and A for Aspergillus fumigatus. All right, coccidis, pneumocystis, Aspergillus fumigatus. All of these stain silver, and the coccidioids, these fungi are stained silver right here in this diagram. Fluorescent antibody stain is used to stain many bacteria, viruses, for example, pneumocystis again, Giardia, and Cryptosporidium. Example is FTA-ABS for syphilis. Okay, so what is FTA-ABS for syphilis? Um, it is a fluorescent reproducible antibody absorption test. Is a highly specific confirmatory blood test used to detect antibodies against Treponema pallidum, the bacterium that causes syphilis. Okay, so fluorescent antibody stain, FDA is fluorescent antibody stain for syphilis, and is also used to detect pneumocystis, giardia, and cryptosporidium. Next, we have special culture requirements. Okay, so these are the bugs that use a specific media for their isolation, and these media have specific contents or other notes that are in this column. The first is H. influenza. H. influenza uses chocolate agar as a medium for isolation. Okay, first of all, we're going to see what agar is. Not the video. Oh no, agar. Not agar. Okay, so it is a jelly-like substance derived from red algae, primarily used as a culture medium in microbiology to grow microorganisms. It consists of polysaccharides and is commonly found in petri dishes. Okay, so there are polysaccharides that compose the agar, and it is derived from red algae. So the media used for H. influenza is going to be chocolate agar, and the factors 5 and 10 in Latin are the contents of this chocolate agar. Okay. So factor 5 and 10 and 19 are the components of this chocolate agar. Then we have Neisseria gonorrhea and Neisseria meningitis. They are going to use the media chosen as here Martin agar. It selectively favors the growth of Neisseria by inhibiting growth of gram-positive organisms with vancomycin, gram-negative organisms except Neisseria with trimethoprim and colistin, and fungi with nystatin. Nystatin. Very typically cultures Neisseria. Very typically cultures in Syria. Okay, so what is a Martin agar? This is my first time hearing about the Martin agar. It is a selective culture medium. Okay, that much I know. It inhibits the growth of other bacteria and fungi that may be present in clinical samples. And this selectivity is crucial because in this area are often found in specimens that contain mixed microbial flora, such as urogenital secretions or pharyngeal swabs, as these pathogens have complex nutritional requirements that prevent them from growing on standard blood agar. Okay, so I want to see an image. This is what it looks like. This is in the chocolate medium, and this is a thin Martin medium for this area. Okay. So it's selectively favored in this area by preventing the growth of positive organisms with vancomycin. This one has vancomycin in it, and gram-negative organisms except Neisseria with trimethoprim and colistin, and fungi with nystatin. So basically, the composition of it is vancomycin, this antibiotic, trimethoprim, and colistin for inhibiting other negative organisms, and for inhibiting fungi, there is nystatin. Next, we have Bordetella pertussis, which uses Gango agar, bodied for Bordetella, and vegan lowe medium. These are composed of potato extract, this bodied Gango agar, and this vegan lowe medium is composed of charcoal, blood, and antibiotic. Okay, so blood, antibiotic, and charcoal, vegan lowe medium. I want to see vegan lowe medium as well. Vegan lowe medium. Yeah. They are made of charcoal, blood, and antibiotic, as given by their black appearance. Next, we have M. tuber, okay, C. diphtheria, which requires a telluride agar or a Loeffler medium. Telluride agar. Uh, it looks like black on the back side of blood agar. So telluride, the word telluride means telluride agar. Um, Selective and differential medium primarily used for isolating Corynebacterium species, especially Corynebacterium diphtheria, by exploiting their ability to produce potassium telluride to produce gray-black colonies. So yes, Corynebacterium diphtheria can cause potassium telluride to be reduced, and produce gray black colonies on blood agar. So Telluride agar is specifically being used for cornybacterium, diphtheria, and Loeffler medium as well. And what might Loeffler medium be? It's a specialized cultural medium primarily used for the cultivation of cornybacterium. It contains horse serum, beef extract, dextrose, and proteose peptones, which provide essential nutrients for bacterial growth. This medium enhances the formation of metachromatic granules, characteristic of C. diphtheria, and is effective in restoring the virulence and identifying properties of fastidious pathogenic or microorganisms. Okay, and then Mycobacterium tuberculosis. It requires a Lowenstein-Jensen medium, middle brook medium, and a rapid automated broth cultures. Lowenstein-Jensen, middle brook, and rapid automated broth cultures. So, a Lowenstein-Jensen. I'm going to see it first. Lowenstein-Jensen. Okay, it looks very green, a new fresh color. Lowenstein-Jensen. And we have middle brook medium. Right there, middle brook. Okay, this one looks quite transparent and not as much as in news as well. So M tuberculosis requires a Lewin-Johnson medium, a middle brook medium, and rapid automated blood cultures. Mycoplasma pneumoniae requires Eaton agar, and Eaton agar requires cholesterol in itself. Lactose fermenting enterics require McConkey's agar, and this fermentation produces acid, causing colonies to turn pink. Okay. The lactose fermenting enterics cause the fermentation in McConkey's agar and produce an acid that causes the colonies to turn pink. Then we have E. coli that requires an eosine methylene blue agar, EMB agar, colonies with green metallic sheen. So an EMB agar has colonies with green metallic sheen. Uh, EMB agar. Mm. Okay, it is basically used to distinguish lactose fermenters from non-fermenters, and here it is a green sheen. Green metallic sheen. Then we have Brucella, Francisella, Legionella, and Pasturella. These names are so, they give me the vibe of Whimsy Princess and her sisters. Buffered charcoal yeast extract agar with cysteine and iron. See, they have special requirements. Buffered charcoal yeast extract agar with cysteine and iron. The Ella siblings. Bruce, oh, so yeah, they do get the feeling of siblings. Bruce, Francis, a legionnaire, and a pastor built the cysteine chapel out of charcoal and iron. Oh, that's such a good morning. And then we have fungi that requires a separate agar. Sab's the fun guy. Reviewing all these, chocolate agar was required by H. influenza, Sir Martin agar by Neisseria, and it had vancomycin, trimethoprim, and cholecystine and nystatin. Then we have Bordetella by Bordet, obviously, and Regal Lower Medium, which had a black appearance, charcoal blood and antibiotics. C. diphtheria by Tellerite, Agar, and Loeffler Medium. M. tuberculosis with Lowenstein, Jensen, and Middle Group. M. pneumonia with Ecometer that requires cholesterol. Lactose fermenting with Meconic, non-fermenting with EMBs. These siblings with buffered charcoal yeast requiring cysteine and iron, and fungi with several agar. What might be confusing would be Bordetella pertussis having regal lower medium, Corynebacterium diphtheria having these two, Tellerite and Loeffler, and M. tuberculosis having Lowenstein Jensen or Middlebrook medium. The rest are quite easy to remember, at least for me. And lactose fermenting require McConkey again, and E. coli, which is non-fermenting, requires EMB. And these siblings require they have a big wish. Buffered charcoal yeast extract agar with cysteine and iron, and fungi requires seborrheic agar. Next, we have anaerobes. Um, I hope we are not. Um, been recording for a long while now, but um, there is a lot left right now. So I think we'll do two more pages and then in the next video. Okay, so anaerobes examples include clostridioids, Clostridium, bacteroids, Fusobacterium, and Actinomyces israelii. Okay, so there is bacteroids, Fusobacterium, Clostridioids, and Clostridium. Iodes, and Fusobacterium and Actinomyces. They lack catalase or superoxide dismutase and are thus susceptible to oxidative damage. Okay. Because both of these enzymes could convert the oxygen radicals produced into a less harmful substance, but they lack them, so they are susceptible to oxidative damage. They have generally a foul smell. Short chain fatty acids cause this foul smell, and they're difficult to culture and produce gas in the tissue, for example, carbon dioxide and hydrogen. Anaerobes cannot breathe fresh air. Anaerobes are normal microbiota in the GI tract. They're typically pathogenic elsewhere. Example causes aspiration pneumonia. Aminoglycosides are ineffective against anaerobes because these antibiotics require oxygen to enter into bacterial cell. Okay, so again, it can't breathe fresh air. Clostridium, Clostridioids, um, bacteroids, um, Fusobacterium and Actinomyces. And aminoglycosides are ineffective against these because these enzymes require antibiotics. Then we have facultative anaerobes. They may use oxygen as a terminal electron acceptor to generate ATP, but can also use fermentation and other oxygen-independent pathways. So these include streptococci, staphylococci, and enteric gram-negative bacteria. Okay, and next we have intracellular bacteria. These are obligate intracellular and facultative intracellular. So the obligate intracellular have rickettsia, Chlamydia, Coxiella, and they rely on host ATP. They stay inside when it's really chilly and cold. And the facultative intracellular are Salmonella, Neisseria, Brucella, Mycobacterium, Listeria, Francisella, Legionella, and Yersinia pestis. Again, that sister right here. The two sisters. Some nasty bugs may live facultatively. Okay, these mnemonics, I would forget them in a minute. But again, the obligate intracellular, these are those bacteria that live inside and they cannot live inside. The facultative intracellular are those that can produce their own nutrients and metabolic machinery to survive outside, but they prefer to stay inside and leech off the cellular organism. So these are Salmonella, Neisseria, Brucella. Again, that sister. These sisters, they are very entitled. And Listeria, Mycobacterium, Yersinia pestis, and of course the sisters Francisella and Legionella. And some nasty bugs may live facultatively. Then we have encapsulated bacteria. The example are Pseudomonas aeruginosa. Okay, we've done these. I've done these somewhere before because I remember these signs. My sky is demonic. But they're covered by a capsule and they are Pseudomonas aeruginosa. Please shine Streptococcus pneumoniae, which is also presented here. Hemophilus influenza type B, H in the shine. Then we have Neisseria meningitis, Escherichia coli, Salmonella, Klebsiella, and group B strep. The capsules serve as anti-phagocytic virulence factor. The capsule polysaccharides, positive or negative protein conjugate, can serve as an antigen in vaccines. I have discussed that before. A polysaccharide antigen alone cannot be used to present T cells. Immunogenicity can be enhanced by conjugating capsule antigens to a carrier protein. Okay, so what they're saying is that the polysaccharide antigen cannot be alone presented to T cells. When we have to enhance the immunogenicity of these organisms, we do so by conjugating these capsule antigens to a carrier protein and then presenting it to T cells. They're opsonized and then cleared by spleen. These encapsulated bacteria. Asplenics have decreased opsonization ability and this increased risk for severe infection. Yes, I've studied these in immunology. And they need vaccines to protect against NSH, and meningitis, S pneumonia, and H influenza. All these encapsulated bacteria require vaccines because in asplenics because they cannot be opsonized and cleared. Then we have the urease positive organisms, P. chunks, Proteus, Cryptococcus, H.
pylori, ureaplasma, nocardia, klebsiella, S. epidermidis, S. saprophyticus. The urease hydrolyzes urea to release ammonia and carbon dioxide, and that increases the pH of the medium. It predisposes to steroid or magnesium ammonium phosphate stones, particularly protease. This is an important clinical aspect that the urease positive organism, especially protease, causes steroid or magnesium ammonium phosphate stones in the kidney. And the bacteria again are protease, Cryptococcus, H. pylori, ureaplasma, nocardia, klebsiella, and both the Staph epidermidis and Staph epidermidis. Next, we have catalyst positive organisms. Catalyst degrades H2O2 into water and bubbles of oxygen before it can be converted to microbicidal products by the enzyme myeloperoxidase. People with CGD, NADPH oxidase deficiency, have recurrent infections with certain catalase positive organisms. Catalase positive organisms include Candida, Pseudomonas, Nocardia, Bordetella, Burkholderia, Capacea, Helicobacter pylori, Aspergillus, Staphylococcus, Serratia, Listeria, E. coli, and the mnemonic is notoriously big bubbling vessel. Notorious for, okay, the first catalase positive, Candida, Pseudomonas, Nocardia, big for Bordetella, and then for Burkholderia, Helicobacter pylori, Aspergillus, Staph, Serratia, Listeria, and E. coli. These are so difficult to remember. They must be. The pigment-producing bacteria are Actinomyces israeli that produces the yellow sulfur granules, which are composed of filaments of bacteria. And Israeli has yellow sand. And then we have Staph aureus that produces a golden yellow pigment. And the aureus is for gold. Yes, I know that one because I've done the sketchup for this one. So it says that Steph is golden yellow pigment. Then we have P. aeruginosa that has a blue-green pigment, pyocyanin and pyovidin. Arugula is green. Then we have Serratia marcinus that has a red pigment. Think red sriracha hot sauce. Okay, Serratia for sriracha. And then we have in vivo biofilm producing bacteria, including Staph epidermidis, Viridans streptococci, P. aeruginosa again, and non-typable and unencapsulated influenza. S. epidermidis causes biofilm to adhere to catheter and prosthetic devices infections. Streptococci viridans causes dental plaques and infective endocarditis. P. aeruginosa causes respiratory tree colonization in patients with cystic fibrosis and ventilator-associated pneumonia and contact lens-associated keratitis. And then again, this non-typable influenza causes otitis media. We now have spore-forming bacteria, and these are some gram-positive bacteria that can form spores when nutrients are limited. Spores lack metabolic activity and are highly resistant to heat and chemicals. Spore contains dipichloropolinic acid, responsible for heat resistance. Most must autoclave to kill spores as is to surgical equipment by steaming at 121 degrees Celsius for 15 minutes at a pressure of 50 psi. Hydrogen peroxide and iodine-based agents are also sporicidal. So the examples are B. anthracis that causes anthrax, B. cereus that causes food poisoning, Clostridium botulinum that causes botulism, Clostridioides difficile that causes pseudomembranous colitis. What is pseudomembranous colitis? Pseudomembranous colitis. This is where inflammation of colon, most commonly caused by Clostridioides difficile infection, often following antibiotic use. Okay, so this is a pseudomembranous colitis that occurs in the intestine. It is because of Clostridioides, Clostridioides difficile. And Clostridium perfringens causes gas gangrene. Clostridium tetani causes tetanus. And all these Clostridia and these bacilli can produce spores. Autoclave to kill Bacillus and Clostridium ABC. And then we have bacterial virulence factors. These promote evasion of host immune response. The first is capsidopolysaccharide, highly charged hydrophilic structure, acts as a barrier to phagocytosis and complement-mediated lysis, major determinant of virulence. As we've also discussed before in the cell wall, the capsule is highly charged hydrophilic structure that acts as a barrier to phagocytosis. Then we have protein A that binds Fc region of IgG. It prevents opsonization and phagocytosis, and it is expressed by Staph aureus. Then we have IgA protease, that is an enzyme that leaves IgA, allowing bacteria to adhere and colonize mucous membranes, secreted by S. pneumonia, H. influenza, type B, and Neisseria, Shin. NSH again, S. pneumonia, influenza type B, and Neisseria. They secrete IgA protease. And then we have M protein. It helps prevent phagocytosis, expressed by group A strep, and the sequence homology with human cardiac myosin possibly underlies the autoimmune response in an acute rheumatic fever. Okay, so acute rheumatic fever is caused by an autoimmune response that is because of M protein. And it is expressed by group A strep and it sequences homology with human cardiac myosin. Um, okay, so the sequence of this M protein is similar to cardiac myosin, and this is a molecular mimic of the body. We will discuss further in the next video. This was it for this one. Thank you so much for watching. Bye.